Welcome to the Unlocked Podcast, a podcast where we decode human performance one gene at a time. I'm Tony, and this series isn't just about science. It's about unlocking the hidden power you carry in your biology so you can train smarter, live longer, and activate what most people think is fixed. We're going to talk about genes, healing, mental clarity, recovery, and even where science and soul start to meet. But we're starting with the foundation because most people don't know what DNA really is or how much control they actually have. Hey, what's up, everybody? Thank you for listening. As always, thank you for coming back. I just wanted to let you know before we start, this is going to be a 2-part episode with the actionable steps really outlined in part 2. So you can go ahead and skip to part 2. But if you want to get the full idea and full understanding of methylation of the MTHFR gene and homocysteine, go ahead and listen all the way through. Enjoy. If you've ever seen MTHFR on a DNA report or a lab printout and immediately started connecting the dots to every symptom you've ever had, I get it. Most of the stress here comes from scale. One gene gets treated like the whole engine, and then everything starts sounding bigger than it actually is. MTHFR, I like to call it the mother gene, but you guys can say all of the letters, whichever feels more comfortable for you. MTHFR lives inside folate biology. Folate is a B vitamin, meaning a nutrient that your cells use for core chemistry, especially when they're building DNA during cell division and making new blood cells in the bone marrow. If you picture the body like a construction site, folate is part of the supply line. When the supply line gets shaky, the places doing the most building tend to feel it first. Here's a quick fun fact that actually helps us stick. The word folate comes from the Latin word folium, meaning leaf, because leafy greens were one of the earliest places it was identified. Small detail, but it grounds the whole topic. This is not mystical. It's the kind of biology that you can literally eat. You know I love my history, so I'm going to tell you a little bit about it. As scientists mapped more of folate chemistry, they described a larger network often called one-carbon metabolism, which is a connected set of reactions that move tiny one-carbon units around to support jobs like DNA building and methylation. Methylation is a chemical transfer where a methyl group, 1 carbon, and 3 hydrogens get attached to another molecule. And that little tag can change how a molecule behaves without changing what it is at its core. Think about it like it's a neighborhood. MTHFR is one address on the entire street. MTHFR stands for methylene tetrahydrofolate reductase, and it codes for an enzyme, a protein that helps a chemical reaction happen fast. In simple terms, it helps convert one form of folate into another form the body uses to recycle homocysteine back into methionine. Methionine is an amino acid, a protein building block. Homocysteine, is part of the same loop. It's an in-between step that's supposed to keep moving rather than build up. And this is where people miss the bigger picture because the recycling step needs more than one ingredient. That ingredient is vitamin B12. Vitamin B12 is one of the key partners. B12 acts as a cofactor, a helper molecule for methionine synthase, the enzyme that actually converts homocysteine into methionine. So, MTHFR helps supply the right folate form, and B12 helps the conversion step run, which is why this never makes sense as a one-gene conversion. It's a pathway, and pathways have partners. Here's a picture to help keep this human. Imagine a recycling line where materials are supposed to pass through stations and keep moving. Homocysteine is like one of those in-between items. If it starts backing up, that's information, not really its identity. It can be a signal that the flow is under-supported or that the system is carrying more load than usual. That marker is why homocysteine became a popular biomarker. A biomarker is a measurable signal, often in blood, that can correlate with what the body is doing. The part people forget is that a biomarker can be meaningful without being the master lever. It can be a clue without being the cause. This is where genetics get talked about the most. One common MTHFR variant is called C677T. Now, I like to always define a lot of these terms because for the person that's listening, they're not going to really understand a lot of this science jargon. So I always leave the definitions at the bottom so everyone can understand them. I explain them inside of the podcast, but if you would always like to get a deeper understanding, feel free to check that out. So let's continue. Variant just means a common DNA difference across the population. I don't know if you guys ever heard of heterochromia, but when you get some time, check that out. My son did mention it to me, and it's like a variant or genetic difference in the eye. Maybe that can give you a little bit more ground of what I'm getting into. Anyway, forget my tangent. Many studies report lower average enzyme activity with this variant, especially when someone has 2 copies. Lower average activity does not automatically mean dysfunction in every person. It usually means the pathway may have less margin under certain conditions. And one of those biggest conditions is low folate status. So once you understand what the pathway is doing, the next thing that matters is how we know what matters in real life. A lot of confusion happens when we treat every kind of evidence like it carries the same weight because a pattern in a population and a result from a trial are not the same kind of signal. In population studies, meaning studies that observe patterns across lots of people without assigning an intervention, higher homocysteine has often been associated with higher cardiovascular risk. Association just means the two travel together in data. It can be a useful clue, but it does not prove that one is causing the other. That clue is exactly why researchers tried the obvious next move. If homocysteine is part of the story, what happens when we lower it with folate and B vitamins? When you test that in randomized controlled trials, meaning studies where people are assigned interventions by chance so we can test cause more directly, the homocysteine number can move, but the clinical outcomes people care about do not consistently improve across large trials. That's the important lesson. Sometimes the dashboard light is real, but changing the light doesn't necessarily change the engine. That same lesson is why the word methylation gets inflated so easy. Methylation is real chemistry, but people often talk about it like it's a master switch for your whole life, and that is where the scale gets lost. DNA methylation is one form of methylation where small chemical tasks can influence gene expression, meaning how actively a gene is used to make proteins, and it behaves more like a dimmer than a switch. It can nudge patterns over time, but it doesn't flip your biology into a new personality overnight. MTHFR sits in a folate-related corner of that landscape, which makes it relevant, but it still does not run the entire methylation system, and it does not single-handedly explain mood, attention, or personality. When someone tries to sell you that story, they're usually taking one piece of biology and stretching it past what it can actually support. One newer research approach is trying to answer the causality question more cleanly by using genetics as a kind of natural randomizer. That's what Mendelian randomization is aiming to do. It uses genetic variants like a natural experiment to estimate whether a biomarker is more likely causal or mainly correlated. What makes it useful is that it often shows the picture is not uniform across outcomes, which supports the posture we're building here. Keep claims tied to the specific outcome and keep confidence proportional to the evidence. So here's what you're going to do with all this in real life without spiraling or going crazy. If MTHFR shows up on your report, treat it like a context. Again, not a verdict. It is not the end-all be-all because a single variant is never a complete story. And if you actually want to evaluate the pathway, don't start with assumptions. Start with signals you can check. Homocysteine is one of those signals, and it becomes much more informative when you look at it alongside folate and B12 status instead of staring at the gene by itself. If your basics are wobbly, you got to stabilize them first because sleep consistency, food consistency, and correcting clear deficiencies usually move the needle more than stacking interventions because of one variant. If you want a clean recap to keep in your head, it's this. MTHFR is an enzyme in the folate network. A common variant can lower average activity, and that can be associated with higher homocysteine in some contexts, especially when folate is low. Homocysteine can be a useful signal, but lowering it does not automatically prevent disease. And methylation is real chemistry. But MTHFR is one part of the landscape, not the whole story. If you guys don't want to say MTHFR, I like saying mother gene. I think people do kind of refer to it like that. But that is a personal preference. Stay tuned for part 2. And guys, as always, remember, your biology listens. Live like it.